Welcome to this explainer. We're skipping the fluff today and diving straight into exactly what Cambridge examiners are looking for in your 1.2 lysosomes answers. We're tackling a classic exam scenario where just one tiny mistake, often made in the very first few sentences, costs students massive marks and absolutely wrecks their grade boundaries. So if you've ever felt like you completely knew the biology but still somehow dropped points on the exam, well, you're in exactly the right place.
Take a look at this comparison. On the left, we've got a flawed generic answer. This is the kind of mistake that just hemorrhages marks. But on the right, you'll see the improved, top band, context-specific phrasing that literally locks in your grade. The generic side just lists basic facts, right? Saying things like, lysosomes contain hydrolytic enzymes for digestion. I mean, sure, that's biologically true, but your ordinary revision completely fails here because it doesn't actually answer the specific question being asked. The top band answer, on the other hand, takes that same concept and links the lysosome action directly to the evidence and data provided in the specific scenario. Honestly, this is the exact difference between a student who merely memorized the textbook and a student who truly understands how to play the exam game.
So let's talk about the method mark strategy. This is how you actually rack up those points by showing your entire process. Now, in a bit, I'm going to reveal an examiner clue that will change your answers completely, but first, you really need the framework that makes that clue obvious. The method mark strategy simply means examiners are awarding you marks for demonstrating the correct analytical process and the procedural sequence. Basically, they want to see your scientific reasoning step by step. You don't just jump straight to the final conclusion. You have to earn points for the journey itself.
All right, let's move into Section 1, Core Biological Principles.
As you can see here, examiners heavily prioritize analytical depth and connected conceptual understanding over just superficial breadth. Top-performing students don't just memorize a bunch of isolated facts. They actually show how these concepts connect. The examiners, they don't want a brain dump of every single thing you know about lysosomes. What they want to see is your logical reasoning applied to brand new, unseen experimental setups. Which brings us to Section 2, the Technical Methodology Framework. This gives us a standardized step-by-step process that you absolutely must follow in your answers. First up, identify the key elements in the practical investigation. Second, apply the relevant principles, things like the role of hydrolytic enzymes. Third, execute your analysis with clear, highly visible working. Fourth, verify your results by double-checking for any anomalies. And finally, Step 5, present your findings in an appropriate format, like a well-structured data table or a graph. Listen, skipping any of these steps is exactly how you lose those absolutely crucial method marks.
Next up is Section 3, Essential Statistical Analysis, where we choose the right test. This table right here simplifies the whole shebang. It breaks down exactly when to use a t-test, chi-squared, or a correlation test, which guarantees you aren't dropping easy methodology marks. Think about it. If you're comparing the means of two different sets of lysosome activity data, you're going to use the t-test. Dealing with categorical data, that's chi-squared for sure. And if you're looking for relationships between variables, like say enzyme concentration versus the rate of digestion, you'd hit that with a correlation test. Honestly, lock this table into your memory and you're going to navigate those data interpretation sections with absolute confidence.
Now, for Section 4, Critical Examiner Warnings. Let's look at some major exam mistakes.
So these are the top 3 traps you absolutely must avoid. Number 1, confusing cause and effect in lysosome action. Examiners frequently report that students just completely mix up the sequence of events during phagocytosis and digestion. Number 2, forgetting to check your units and terminology. Never, ever leave your data naked. Always include the specific units. And number 3, providing answers that aren't specific to the context given. Actually, let me show you exactly what this third trap looks like in practice.
I want you to pause for just a second and really think about this scenario. If an exam question asks you about lysosome activity in a specific white blood cell that's fighting a pathogen, but your answer just describes general cellular digestion without mentioning that pathogen or that specific cell type, what happens? Well, you lose the marks. It's that simple. Your perfectly accurate biological description totally fails because it isn't anchored to the actual prompt.
So the takeaway here is absolutely crucial. You must always, always anchor your lysosome knowledge to the exact context provided in the exam prompt. Consider this an official examiner alert. The classic mistake is providing generic answers. If the exam gives you a case study, your concept, your data extraction, and your scientific reasoning must all explicitly reference that specific case study. No exceptions.
Moving right along to Section 5, the final evaluation strategy for higher order thinking.
If you want to reach that top evaluative level, your answers need to systematically hit every single one of these points. First, you've got to identify limitations and assumptions in the experimental design. Then, you need to consider alternative interpretations of the data. When they ask you to compare, make sure you identify both similarities and differences using clear comparative connectives. And you have to support each of those comparisons with hard evidence. Finally, wrap it all up by drawing justified conclusions.
As this quote points out, examiners explicitly note that top-scoring candidates demonstrate evaluative thinking by questioning assumptions and justifying their decisions with actual evidence. You aren't just a robot reciting facts. You're a scientist evaluating the real quality of the evidence in front of you. But here is the massive unresolved problem we've been building towards this whole time. Even if you completely master this entire framework, you nail the method marks, the statistical tests, the contextual anchoring, and the evaluative thinking, there's still one hidden examiner clue that can change your answer completely. If you miss this one tiny detail, your perfectly structured answer will get severely capped. And honestly, you won't even realize why. So, do you know what it is? That hidden cause is the exact command word used in the question. Simple words like explain, describe, or evaluate completely change the mark scheme for the exact same biological topic. It's wild. We're going to decode every single one of these in our next mandatory explainer so you can fully unlock your highest possible mark band. You really can't afford to apply today's framework without knowing exactly how those command words direct it. So make sure to subscribe to our channel, Ace the Exams, to truly excel in your Cambridge exams. Go ahead and click the explainer on your screen right now to master those command words, and I will see you over there.